Every time a person steps into a room, they leave behind a microscopic ID card that they didn't even know they were carrying. Today, we're cracking open the ultimate biological blueprint, DNA. From a single strand of hair to a smudge on a doorknob, we're exploring how forensic science turns invisible crumbs of code into the keys that unlock the answers into sometimes decades-old cold cases.
Come take a deep dive with us into the world of DNA. You're listening to Lurking in the Shadows.
All right, welcome, Shadows.
Hello, everybody.
Welcome back, or welcome if this is your first episode with us.
If this is your first episode, you picked a unique one, I'll say.
You did. This is our 1-year anniversary episode.
Whoa!
Woo!
We did it!
Happy 1 year to our podcast.
Yeah, here. Cheers, Kirsten.
Cheers.
Yeah. Bing!
Cheers with us.
Yeah.
Get yourself a drink. And you might need one. This is going to be a long episode.
Yeah, you might literally just get thirsty.
Yeah.
We are going to probably throw a lot of information at you, but we will try and keep it interesting, I will say. But I think DNA is quite interesting. So
it is, yeah. And as you heard from the intro, we're doing a DNA deep dive because there's many cases that we cover that have been solved with DNA, mostly in our news episodes. But one of our slogans has kind of become "Hell yeah, DNA."
Yeah. We
so we figured this is a good time to talk about it.
Yeah. Yes, and I think a lot of the news cases come up because how much DNA and forensic science has developed nowadays, and that's why we get to talk about those cases. And I think in the end, past all the science mumbo jumbo that we're going to throw at you, in the end, DNA is cool as shit. And it's making such good leaps and bounds in solving cases that, like, we should be talking about it. Like, how cool is this?
Yeah. So nerd out with us today as we go into, like, the basics of DNA. The beginning of this episode, stick with us because the beginning is going to be a little bit of a bland science lesson. But if you like true crime, you should appreciate DNA.
Yeah, and we're going to go over how it plays into solving cases and one of the first cases that was solved by DNA.
Yeah. And then we're also going to show or talk about a few cases that have been solved through genetic genealogy as well.
Yeah. So, hell yeah, DNA.
Hell yeah, DNA. All right. So what is DNA? DNA is deoxyribonucleic acid, and it is the molecule that carries genetic information for the development and functioning of an organism. DNA is the set of instructions that make each person unique. These instructions tell your body how to create organs, tissues, how to function, and even what you look like. All living things have DNA in their cells. You inherit your DNA from your biological parents. You get half from your mother and half from your father. Most DNA is located in the nucleus of a cell, which is also called nuclear DNA. But a small amount of DNA can also be found in the mitochondria, where it is called mitochondrial DNA or mtDNA, which is usually only inherited from your mother. And you should remember from 7th grade science class that mitochondria are the powerhouses of the cell.
You're rattling.
That is one lesson. Yeah, as one lesson, like I literally, before we even started this, I told Brandon, mitochondria is the powerhouse of the cell. Like I just remember that in my brain.
In your brain. Yeah, the weird things that you remember from science.
Yeah. So mitochondria are organelles within cells that convert glucose and oxygen into adenosine triphosphate, or ATP, which is energy that a cell needs to carry out all of its functions. Your DNA as a whole, with all these things included, makes up your genome. So DNA is the chemical molecule that carries genetic instructions, while the genome is the entire collection of an organism's DNA, including all its genes, chromosomes, and non-coding DNA. And if you don't remember, a chromosome is a large structure made of tightly packed DNA and proteins Think of chromosomes like a book, while a gene is a specific segment of DNA within that chromosome, like a chapter in a book, and it contains the code for a particular trait or protein such as eye color or hair type. So the chromosome organizes, packages, and carries the DNA, while genes are the functional units of heredity located on those chromosomes. So an interesting analogy I found is you can think of your genome as a whole as a library. Chromosomes are the books within that library, and genes are the chapters of each book.
I like that. That's a good way to put it.
Yeah. So now you might be asking, well, what does DNA actually do? DNA carries the genetic instructions required for development, growth, reproduction, and daily functioning. It organizes these instructions into genes, which control the creation of proteins that perform vital cellular functions. Human DNA consists of about 3 billion bases, and more than 99% of those bases are the same in all people, which is kind of crazy.
That is, I actually read that fact in my research, and yeah, that blew my mind.
Yeah. Like 99% of us were the same. That's crazy.
We're the same. Yeah, that's
Yeah.
crazy.
DNA bases are the 4 chemical components, adenine, cytosine, guanine, and thymine, that store genetic information. The sequence of these bases determines the information available for building and maintaining an organism. DNA is essentially the instruction manual for life, written in a 4-letter chemical alphabet. It holds the instructions for everything about you and contains the code for cells to read to make proteins, which proteins are the molecules that perform most bodily functions. There can
That's
also
why
be
protein's
errors in your
important.
DNA. Yes, very. Errors or damage to your DNA can lead to health issues. DNA has the ability to proofread its sequence to make sure there aren't any errors when it makes more copies of itself, but sometimes the proofreading process can fail, leading to changes or mutations in your DNA. These
Ah,
mutations can cause cancer, lung disease, heart disease, diseases in other organs, can cause skin changes and premature aging, fertility issues, and congenital conditions, which are conditions present at birth.
like my birth defect.
Yes, it was
Yes.
probably due
So,
to an error in DNA replicating.
yeah, in the process of probably my body developing my eye, it would
Mm-hmm.
have failed somewhere along that way.
Yeah. Mhm. So, what is the shape of DNA? DNA is made of 2 linked strands that wind around each other to resemble a twisted ladder, a shape known as a double helix. Each strand has a backbone made of alternating sugar, which is deoxyribose, and phosphate groups, with the base pairs forming the ladder's rungs and the sugar and phosphate molecules forming the vertical side pieces of the ladder. The information in DNA is stored as a code made up of 4 chemical bases, like I mentioned, adenine, guanine, cytosine, and thymine. DNA bases pair up with each other. A pairs with T and C pairs with G to form units called base pairs. The sequence of the bases along DNA's backbone encodes biological information, such as the instructions for making a protein or RNA molecule, like we discussed. So, how was the shape of DNA discovered? Well, that brings us to the next topic of the amazing Rosalind Franklin. Rosalind Elsie Franklin was a brilliant chemist who was born in London on July 25, 1920. She was the second of 5 children in a prominent Anglo-Jewish family. Rosalind attended St. Paul's School for Girls, which was big on preparing its graduates for careers, not just marriage.
Hell
So I
yeah.
feel like that school was kind of ahead of its time
Hell
because
yeah.
back then women were expected to just be homemakers.
Yep.
Rosalind had taken a liking for math and science and easily learned other languages. She would eventually speak excellent French, good Italian, and passable German.
Wow. Brilliant.
Yeah, yeah. I'm still struggling with my Spanish from high
Yeah,
school.
me too. somebody somebody called at work the other day, and I tried so hard, and I always want to try. I always want to try.
Mhm.
I want to
Yeah,
learn. And then it just makes me feel bad that I can't, And so
yeah.
I'm like, I need to practice.
I know. Like I can speak to someone in Spanish a bit, but once they start talking back to me, I'm like, wait, I
I
can't keep
know.
up.
There's no doubt. I can't. My brain.
Yeah. Oh, and speaking of Spanish, Bad Bunny was amazing at the Super Bowl halftime show.
Yeah, a Super Bowl was lame, but yeah.
Her mom recalled, quote, all her life, Rosalind knew exactly where she was going, and at 16, she took science for her subject, end quote. Instead of remaining at the school for girls for an extra year for more college prep, she left St. Paul's in 1938 to enter Newnham College, one of two women's colleges at Cambridge University, where she majored in physical chemistry. Her undergraduate years were disrupted by World War II. Many instructors, especially in the science department, had been pulled into war work. Some faculty, for example, biochemist Max Perutz, were detained as aliens. In one letter, Franklin noted, quote, practically the whole of the Cavendish laboratory have disappeared. Biochemistry was almost entirely run by Germans and may not survive, end quote. Cambridge also took in a number of war refugees, including the French scientist Adrian Whale, who arrived at Newnham in 1940 and became Franklin's mentor and friend. In 1942, Rosalind began work with the recently organized British Coal Utilization Research Association, or BCURA. For the next 4 years, Rosalind worked to explain the microstructures of various coals and carbons and determine why some were more permeable by water, gases, or solvents, and how heating and carbonization affected the permeability. Franklin was the first to identify and measure these microstructures, making it possible to classify coals and predict their performance to a high degree of accuracy. Her work at BCURA resulted in a doctoral thesis and 5 scientific papers, leading to her receiving her PhD from Cambridge in 1945.
Hell yeah,
That
PhD,
is
1945.
wildly impressive.
Yes, that's awesome.
Yeah. Hell yeah, women.
That's right.
After the war, Rosalind followed Adrian Whale back to France, who helped her get a position okay, I'm going to try my best to pronounce this, Jacques Meringue's lab at Laboratoire Central des Services Chimiques d'État in Paris.
You nailed it, man. Just fucking nailed it.
I don't know a single French word, so I don't know, um, the pronunciation on certain letters, but there we go.
No, I don't either.
Here she learned how to analyze carbons using X-ray crystallography, also called X-ray diffraction analysis, becoming very proficient with the technique. Her work detailing the structures of graphitizing and non-graphitizing carbons helped form the basis for the development of carbon fibers and new heat-resistant materials, and earned her an international reputation among coal chemists. Rosalind enjoyed her time in France, but by 1949 wanted to seek work back in England. In 1950, she was awarded a 3-year Turner and Newell Fellowship to work in John T. Randall's biophysics unit at King's College in London. The plan originally was to have Rosalind build up a crystallography section and work on analyzing proteins, but Randall asked Franklin to investigate DNA instead at the insistence of the assistant lab chief Maurice Wilkins, who had begun his own research on the structure of DNA in 1947. Franklin took increasingly clear X-ray diffraction photos of DNA and quickly discovered that there were 2 forms, wet and dry, which produced very different pictures. She discovered the wet form was helical in structure, with the phosphates on the outside of the ribose chains, but her mathematical analyses of the dry form diffractions did not indicate a helical structure, and she spent over a year trying to resolve the discrepancy. By early 1953, she had determined that both forms had 2 helices. So this is when the shape of DNA was discovered by Rosalind Franklin. But 2 men at the Cavendish Laboratory at Cambridge, Francis Crick and James Watson, were also working on a theoretical model of DNA. Unfortunately, in January of 1953, they were given insight about DNA's true structure from one of Rosalind's X-ray diffraction photos, known as Photo 51, shown to them by her lab partner Maurice Wilkins, and from a summary of her unpublished research submitted to the Medical Research Council. They never told Rosalind that they saw her research, then went on to publish an article titled Molecular Structure of Nucleic Acids. Watson and Crick never once touched or looked directly at a fiber of DNA while trying to develop their own model.
Wow.
The article they wrote has no experiments and consists of speculation based primarily on the experimental findings of Rosalind Franklin and Maurice Wilkins' research. Crick later added that Franklin was 2 steps away from realizing the correct structure of DNA in the spring of 1953, not long after they looked at her work. Sadly, by that spring, are we going to say something?
I was just gonna say this is just terrible because they can just like do that and get away with it, especially like in the era. Like anybody would have just looked down at her because she's a woman, even though she literally had a fucking PhD. They would have literally just dismissed any of her findings. Yeah.
Yeah, she was only, like they said, that she was only 2 steps away from the full final structure, and they were able to take all her work and figure out the last 2 steps.
Yeah.
Sadly, by that spring, Franklin had arranged to transfer her fellowship to J.D. Bernal's crystallography lab at Birkbeck College, where she turned her attention to the structure of plant viruses, particularly the tobacco mosaic virus. So she kind of gave up on her DNA research and switched to viruses, not knowing how close she was to that final structure of DNA.
Oh, that's sad.
Her research of the diffraction patterns revealed that the tobacco mosaic virus, or TMV's, genetic material was embedded in the inner wall of its protective protein shell. Her study and expertise in virus structures was recognized by the Royal Institution in 1956, when its director honored her with a request to construct large-scale models of rod-shaped and spherical viruses for the 1958 Brussels World's Fair Science Expedition. Sadly, in the fall of 1956, Franklin was diagnosed with ovarian cancer. She underwent surgeries and treatments, but she died in London on April 16, 1958.
Oh, only 2 years she lived.
Throughout her career, she wrote 19 articles on coals and carbons, 5 on DNA, and 21 on viruses.
Wow. What a
She was very
smarty
dedicated
pants.
to her work.
Yeah.
Crick, Watson, and Wilkins shared the 1962 Nobel Prize for Physiology of Medicine for their work on the structure of DNA, while giving no credit to Rosalind Franklin.
The real person who deserves some credit here.
They won a Nobel Prize for her work.
Yeah. I know somebody else who wants a Nobel Prize.
For doing nothing.
I couldn't help myself. I couldn't help myself. Woo! That was a good one.
No, we just had to throw that in there, you know?
Yes. God. I think she was never given any kind of recognition for it, other than this, what we can tell.
Yeah, not until many years later.
Okay, yeah.
In a footnote to a 1954 paper that Watson and Crick wrote, they stated, quote, "We wish to point out that without this data," referring to Rosalind's research at King's Lab, "the formulation of our structure would have been most unlikely, if not impossible," end quote. So they even admitted, without her work, their journey was impossible.
Impossible, yeah. She was the brains of all this. They literally just put more pieces into place, like...
Yeah. Bitch-ass men.
Yeah, fucking cowards.
In his 1968 memoir titled The Double Helix, Watson referred to Rosalind as Rosie, a, quote, bad-tempered, arrogant blue-stocking who jealously guarded her data from other colleagues, even though she was not competent to interpret it, end quote.
Oh my God. Wait, what year was that?
1968.
I want to know what a blue stocking means. I'm going to Google it. Did you Google it?
Yeah, I don't know. I didn't look that up. An
Is that like a
arrogant blue
harlot?
star
I want to.
must be some English derogatory term.
From the 60s. Um, yeah, a derogatory label of an intellectual or educated woman.
Woman specifically. Wow.
Yes, it was a woman specifically. Yes.
Wow. Wow. Okay.
Yep. Wow. Yeah.
You know what?
God.
Well, then what's the term for an incompetent man? I bet they don't even
From
have
the
one.
No, they didn't entertain something like that.
No.
Yeah, bitches.
And not competent to interpret it. I'm sorry, not competent enough to interpret the data she discovered. The same data you also admitted to likely never discovering yourself. But she's
Yeah,
not
I was gonna say,
competent.
yeah, she wasn't.
Okay.
Yeah, so incompetent. She
Mm-hmm.
she literally
That
gave you
she
everything,
discovered
like I
all
said.
this.
Yeah, egos, man.
Well, yeah. Yep. In 1975, Franklin's friend Anne Sayer published a biography as an angry rebuttal to Watson's incorrect telling of the story, making Rosalind Franklin's true contributions to DNA better known. And then much later on in 1999, Watson admitted, quote, the Franklin photograph was the key event, end quote. There is now that was in 1999. It took him to admit that.
Oh, wow. Wow.
Mm-hmm. There is now a Rosalind Franklin University of Medicine and Science in Chicago. Their website says, quote, our university was dedicated in 2004 to Rosalind Franklin, PhD, the brilliant and trailblazing scientist whose photo 51 revealed the double helix of DNA, a discovery that was essential in unlocking the mystery to how life is passed down from generation to generation. Dr. Franklin's passion for learning, her pursuit of extreme clarity, and her unflinching commitment to the highest standards of scientific research brought lasting benefit to mankind and make her an ideal role model for our students, faculty, and aspiring students for health professionals throughout the world, end quote.
Wow. No.
And then Watson actually just died in November of 2025. He was like
Oh, wow.
97 or something.
Wow. Of course, that son of a bitch made it that long.
Yeah, I know. Do not rest in peace, sir.
Yeah.
And then Crick died back in 2004.
Good riddance. All you had to do was give proper credit. We wouldn't have this attitude. That's all you needed to do. I don't think you're a terrible person, but you did terrible things.
And it wasn't only that they didn't give her credit, they like downplayed her intelligence.
Yeah, also
and called her names.
Yeah. Like you didn't have to do all that, my guy.
Yeah, no. You
we wouldn't be here.
Puny dick bastard.
Yeah. Teeny weeny peeny. I heard that the other day. And I'm taking it. I think it's the I've Had It on YouTube. They're great, Kirsten. You would love them.
I'll check it out.
We will.
So that is that is what is DNA and the history of the DNA shape being discovered.
That was pretty good, actually. That did rattle some things from my high school, I feel like.
Yeah. Yeah, it was, I mean, there was a lot of things. It also brought me back to anatomy and physiology class in nursing school.
Dude, like I haven't done any, yeah, I haven't done anything like as recent as you, but back in school, I was actually I liked both biology and chemistry, but I loved chemistry. I had 104% in chemistry when I took honors.
I did not do well in chemistry because the teacher was no good, and
my teacher was fantastic. It makes a difference. You're right.
Yeah, it does. Yeah, I did not have a good teacher, and he was also not approachable and not nice. And
yeah, I think I was a woman, and she was wonderful.
I either ended up with a C+ or B- because I just couldn't learn in that environment.
No, man, it was great. I loved it. Biology was cool too. I loved
Yeah, I mean, it was
biology.
interesting. I probably would have done better had I had a different teacher.
Yeah, probably. Physics is where I fell, and that's not what we're covering.
No, and we will not.
No. So now it's my little part, right?
Yes.
So I am jumping in to my part of the DNA, which is a little bit more of the forensics part of it. And we're going to take it back to 1970s. It took until the 1970s for DNA to be used to identify people, but it was Jeffrey Glasberg in 1983 who filed for a patent for the use of DNA variation in forensics, specifically fingerprinting. This is based on his work at Rockefeller University. This patent wasn't given until 1997. However, in 1984, an independent British these words are hard.
I know. A lot of big words in this episode.
Yeah, there's going to be some big words in this episode. Geneticists developed a different way to DNA profile individuals. His name was Sir Alec Jeffrey. He made his discovery while working at the University of Leicester, working in the genetics department. He discovered while examining DNA, you can see patterns of similar traits. These traits can also be inherited. This discovery led to the first solved criminal case using DNA profiling. Think of a DNA sample like a needle in a haystack. When a lab gets a swab of saliva, the DNA is actually just a tiny fraction of that sample. Kirsten did great explaining it. The rest is made up of proteins, fats, and cellular debris. The first step of any profile is a process called the extraction. Essentially, the scientist has to pop out the cell, open it up like a balloon, and once the DNA is released, they wash away all of the different junk that's around it that they don't need, purifying the sample.
What an interesting job that would be.
That would be cool. I'd do that. Even if I
Just
couldn't
like
even
breaking open cells and
yeah, like doing that all day. Like I know it takes
Mhm.
I'm taking 2 seconds to tell you how it works. This takes a long time.
Yeah, yeah.
For
And it's
yeah,
sure
for
it's
trying
very tedious work.
yes. So maybe us sitting here saying how cool it is, maybe after doing it, maybe we wouldn't. But I still think I'd be cool just to watch them do it, to be honest.
Yeah, just to like be in a lab for a day and
Yeah,
experience it.
that'd be so cool. Like my
Mhm.
mind would be blown, I think.
Yeah.
So they purify the sample until only the genetic signature remains. It's a delicate process because if you don't get it right, you're just looking at a mess of cellular leftovers instead of a killer's identity. This process can take several days due to how delicate they need to be during each process to preserve the sample. But once we have the DNA sample, there are different ways that we can test it. RELP, or restriction fragment length polymorphism, is a method that cuts the DNA into specific sequences using enzymes. The next technique that is used is called polymerase chain reaction, or PCR, analysis. And the easiest way that I can explain this process is pretty much that they just take the DNA sequence and amplify it, like make it everything as noticeable as possible, making it easier to see the details of the strands. And this
And PCR
practice,
testing is also used in hospitals.
oh, really?
Like when we test for COVID and stuff, and certain viruses, it's called. They use the PCR method.
Oh, you know what? I didn't even make that connection. I've seen that. Yeah, you're right.
Mhm.
Yeah. So it's just like probably what they do with the COVID testing is just like instead of having to send out the lab test, it amplifies your results type of thing, like
Mhm.
whatever
Yep.
it amplifies. Yeah. This practice was developed in 1983 by Carrie Mullis, and it is used today commonly in medical and biological research labs. The most common DNA profiling process used today is called PCR, what we just covered. When we talk about DNA matching, we aren't usually looking at every single gene in a person's body. Instead, forensic science zoom in on a specific corner of your DNA called short tandem repeats, or STRs. There is also a mini STR And this method is used on situations where we have charred bones or very minimal skeletal remains.
Okay.
Imagine your DNA as a long string of letters. In certain spots, the code just starts repeating itself like a pattern. It might go GTA, GATA, GATA, and on repeat. So now here's the key difference. While we all have these patterns in the same spots, the number of times that they repeat varies from person to person. I might have 5 repeats while you might have 12. These specific locations are called, I want to look this up, Loci. It's Loci, right? Yeah.
How's it spelled?
LOCI. It's Loki.
I think so.
Yeah, because I feel like I'm trying to pull on my inner high school knowledge.
Mm-hmm.
And they act like a unique genetic barcode. In the early days, forensic scientists had to use chemical scissors called restriction enzymes to cut the DNA into pieces to see these patterns. It was slow
Wow.
and required a lot of blood. But today we use a much faster method, again, the PCR. So the PCR, which is our main method, is like photocopying. And instead of cutting the DNA, the scientists use PCR to find those repeating sections and make millions of copies of them to amplify them. By measuring the length of each of these copies, they can determine exactly how many repeats you have at each spot. When you compare 13 or 20 of these different spots between a crime scene sample and a suspect, the odds of a random match are literally billions to 1.
Wow, that's crazy technology. What they can do.
Yeah. And like Kirsten said, like this is a tiny little part because we're just looking at
You need a microscope to see this.
yeah, like, and I mean a microscope that most people aren't affording.
Yeah, not just a basic one.
Yeah, yeah, to get to like these levels, yeah.
Mm-hmm.
And that is only also if you have to remember if we have a pure DNA sample to work with, and they're able to extract what they need from it. Before the PCR methods were developed, any degraded DNA samples were virtually useless because of the damage of the cell makes it too difficult to split the cell. So
Yeah, and think
PCR
about cases where they've held onto this DNA sample for 10, 20, 30 years.
Yeah.
It degrades.
Yeah. And then, yeah, which is it's sad to think about too because we are getting to that point of the technology. But yeah, you come across that.
Mm-hmm.
PCR helped make degraded samples easier to extract DNA from. And so another analysis that they use in forensic profiling is the Y chromosome analysis, which is only used for male DNA because only they have the Y chromosome. There's a database called the Y haplotype reference database that was established in 2000. It is worldwide and helps keep data on the male population's DNA. Mitochondrial DNA is another type of DNA that can be found in hair shafts, teeth, and bones. A sample of DNA that is collected that contains more than one individual's DNA is called a mixture. To simplify how to process these samples and give an example how it might go, let's say our sample is from a sexual assault victim. Making these 3 samples from the victim, the consensual partner that this victim has, and the suspect. So when you're looking at all of these samples, they divide them into sample A, sample B, and sample C. The sample A collected is the one that is the hardest to differentiate from the other 2. The B sample may contain some patterns that stand out more than type C. And then type C is the one that is the most discerning patterns. And that's how they will differentiate a mixture. It took until almost 2000 for a DNA database to become a thing. Kevin Miller and John L. Dawson started the first one ever after studying at the Cambridge University. And nowadays, there are multiple DNA databases out there, Some are private, but most are government controlled. The main system most have heard of is the CODIS, which stands for Combined DNA Index System. This system contains over 13 million records and is monitored by the FBI. It is illegal, though, for the FBI to keep your DNA if you are not convicted of a crime. So if a suspect is acquitted and they have no record, their DNA info must be deleted from the database. Mm-hmm. The risk of a false match is 1 in 1 billion, but it gets more complicated if you count identical twins in there. Then the numbers get a little weird. One way this database has helped solve cold cases is using familial DNA, where the sample is entered into the database and a strong match comes up, but not an exact match, meaning that they are likely related to each other. Partial matches is a thing as well, when the repeating pattern only matches one part of the allele. So that is my little bit on DNA and forensic profiling. I hope it was not too complicated. I tried to keep it as simple because Kirsten did a great job covering everything and I didn't want to repeat too much. But now I'd love to share with you guys what I found to be the first case that was solved with DNA, which I don't know if Kirsten, have you heard of this case before? The Pitchfork Murders?
No, I only came across it in the DNA research.
Okay, yeah. No, I had never heard of it either. So this was new to both of us, and maybe it'll be new to you. But definitely, I feel like a very big turning point in criminal history.
Mm-hmm.
So Colin Pitchfork was born in I'm going to say this wrong. Lockensteiner. Oh, I can't. Leicestershire, England, on March 23rd, 1960. I'm so sorry. His parents, Molly Beck and Alwyn Pitchfork, married in July 1950. Colin went to Market Bosworth before going off to Bosworth College in Desford. He was active while in school, joining Newbold Vendors Scout Group, and he earned the Chief Scout Award in 1976. His mother was the scout leader, and he ended up going to the World Scout Jamboree in Norway. Colin had an incident where he was arrested for indecent exposure, but was only referred to therapy as punishment.
Therapy
By
is
1980,
punishment?
Yeah, therapy was punishment for that. By 1981, Colin married a social worker and they moved to the village in Littlethorpe, England. He had a career in baking, working at Hampshire Bakery. He became a very skilled cake decorator after apprenticing at their head office. He will work there from 1976 until he is arrested then in 1987. Just to think. All this I'm going to tell you is just a baker just decorating cakes in the background.
Okay.
His supervisor's statement about him said, quote, "A good worker and timekeeper, but he was moody and he couldn't leave women employees alone. He was always chatting them up," end quote.
Okay.
After marrying in 1977, he again exposed himself to a school-aged girl and he was fined 30 euros. 30 what? Yeah. For after I was like, I forgot where we were for a second. 30 years after pleading guilty at the age of 17.
He's married. He's only 17. Wow.
Yeah, they got married very early. Yeah. 2 years later, yeah. I mean, it's a different time and probably
It was, yeah.
Yeah. Also being like compared to it overseas, I don't know how it was also back then. You know.
Yeah, because I was just picturing like a man in his 30s showing himself to this poor school-age child. So I
No.
guess
Started
I'm glad
young.
he's only 17.
Yeah.
I mean, it's still horrible, but
It's still terrible. I mean, he does it into adulthood, so it
oh, okay,
doesn't
well,
excuse the age at this point.
mm-hmm.
So, 2 years later, his crimes began escalating when he kidnapped and forced a 16-year-old girl into a field where he assaulted her. Just a year later, on February 14th, 1980, at the age of 20, he again exposed himself to 2 teenage girls and was given just a year probation.
Like, dude, what are you doing? No one wants to see that.
I don't understand what are the flashers or whatever they get out of
Yeah,
that.
like what? Yeah, what are you getting out of that? When
Yeah.
people are 99% of the time, the people's reaction is going to be horrified.
Honestly, in this world, I've seen so many. I'd be like, "You know what, dude? Just like go get a life." Like
Like,
I don't want to see your pain. Just go
no,
somewhere.
no one does.
God. From here on, the crimes just kept escalating. On November 21st, 1983, while 15-year-old Linda Mann was walking home from her babysitting job, she decided to take a shortcut home instead of taking her normal route.
Oh,
When she did not return home that night, her family and neighbors spent the entire night searching for her. The next morning, on a footpath called Black Pad, uh, that is not traveled often, the body of Linda was found. She was sexually assaulted and strangled. The police collected semen samples from the scene and did what they could with the technology that they had at the time. They were able to tell it was someone with type A blood, and it had a profile that matched only 10% of males. This
okay,
is
that's kind of impressive for that time.
for that time, yeah. This is as far as they could go with the DNA at this time, so the case remained open with no suspects. 2 years later, even though I believe he was probably doing this more than what we think, but girls were honestly probably too afraid to come forward. Let's be real here. And
Yeah.
it is that time. In October 1985, Colin again sexually assaulted another 16-year-old, and this time it was reported he held a knife to her throat and had a screwdriver he was threatening her with.
Oh.
This next incident that happened is pretty damn scary. It was a Saturday in June 1986. Liz Knight was picked up by Colin. He drove her 40 minutes from Wigston to Great Glen. Liz, of course, probably so scared, tried to grab the steering wheel. And when she did that, Colin's attitude changed from reasonable to not so reasonable. It's a pretty scary situation if you think about it.
Mm-hmm.
He decided to then another switch in him. He decided to drive her home instead of attacking her. But
Interesting.
when he drove her home and pulled up, Colin put his hand on her knee saying, quote, "I haven't hurt you yet."
Yet.
Colin dropped Knight off at her house asking, quote, "How about a good night kiss?" Which she
Yeah.
refused. And Pitchfork then replied, quote, "I bet you would never accept a lift from a stranger again." And that was it.
Is he trying to teach her a lesson? Like,
I don't know, but I wrote it. I was like, what chilling confidence this man has at this point
mm-hmm.
to do that.
Right.
Like
I mean, I bet she never did hitch a ride again after that, but
he was stroking his own ego doing that.
yeah.
Yeah,
Wow.
it's scary.
Mm-hmm.
His last victim was 15-year-old Dawn Ashworth. On July 31st, 1986, she left her house to go visit a friend at their house. She was expected home by 9:30 p.m. When she did not return home that night, her parents immediately called the cops to report their daughter missing. It was 2 days later on another footpath called Ten Pond Lane where the body of Dawn was discovered. She was tucked in the wooded area just off the path. She was beaten, raped, and strangled. This crime was very similar to the murder of Linda Mann, and they also discovered the blood type matched from the semen samples taken from both victims. Both girls also attended Brockington High School. At this time, a 10,000 euro reward was offered for information leading to the arrest of the suspect. The first suspect they came to for these crimes was Richard Buckland. He was a man with a learning disability. The only reason he became a suspect was because he shared details about Dawn's body and confessed to the murder, but didn't confess to the first murder of Linda. He was only 17 at this time. He is never charged with these crimes.
Okay.
By the time we hit 1987, police were focused on the DNA that they had and decided to ask all local men to come in and give a DNA sample to compare to what they already had. The men of the town that refused were then watched a little closer by police.
Okay.
Around 1,000 men came out to give a sample. One of those men to give a sample was 23-year-old Ian Kelly. He gave a sample on January 29th, 1987. The weird thing is, Kelly didn't live in the area that was being investigated and asked to come in to give their sample. So why is Kelly giving a sample? The truth comes out in a pub in 1988. Kelly, who probably had a few too many, was overheard discussing how he gave a blood sample for his colleague named Colin Pitchfork. He agreed to this when Colin approached him offering 200 euros in exchange for the DNA sample. Kelly agreed and used a fake passport pretending to be Colin, and he went and gave his sample. Later, another coworker said he too was approached by Colin about giving a sample, but he declined. Apparently, this conversation that he had with that coworker was overheard by a 28-year-old Jackie Foggin. She overheard this conversation on August 1st, 1987, but did not report it to police until September 18th. The excuse Colin used to get Kelly to do this was he didn't want to be harassed by police with his prior convictions.
Okay.
Kelly is arrested on September 18th and will spend 18 months in jail for giving a fake sample. The next day, Colin Pitchfork is arrested by Detective Mick Thomas. It is reported that Colin's wife tried to attack him when he told her he killed 2 girls. When
Good for her.
Colin, yeah, right? Yeah, fuck him. Punch him in the face.
Mm-hmm.
When Colin was brought in for questioning, he confessed to exposing himself to over 1,000 women and girls.
That's disgusting.
That is so disgusting.
1,000?
Yeah.
It's probably more than that.
Probably. He ain't going to keep count when it's that high.
Yeah.
He said he couldn't control this compulsion that developed in his teens. He did confess to the crimes, but not to the true violence behind them. He also confessed how his crimes progressed from lesser violent assaults to strangling his victims. He claimed he began strangling them to hide his identity, but police believe there was more of a sexual sadism type motive, not the hide your identity type.
Yeah.
And yeah, I can agree with that, especially because of him letting that one victim go, and more than one victim. The way he let Liz Knight go was like so weird. So I think he was just getting confident and getting off on things like that. Like there was, yeah. The trial started in January 1988 and was held at the Leicester Crown Court. Pitchfork was pleading guilty to the murders and rape of both Linda Mann and Dawn Ashworth. He was also being charged with the sexual assault of 2 other victims and also conspiring to prevent the course of justice. The Lord Chief Justice at the time of his sentencing said, quote, "From the point of view of the safety of the public, I doubt if he should ever be released," end quote. After comparing his DNA from the crimes and positively matching them to Pitchfork, there was not much debate for the jury. He received life in prison for the 2 murders, 10 years for each of the 2 rapes, and 3 years for the other 3 charges, including the 2 sexual assaults. And the conspiring to prevent the course of justice also got 3 years. He must serve a minimum term of 30 years. Now, in 2009, an appeal reduced that term to 28 years. Then you're going to get a kick out of all this. Then on April 22nd, 2016, the parole board heard Pitchfork's appeal presenting evidence of his personal improvements. He got a degree and had kept steady working transcribing music and for the Braille. You know, he's just a wonderful person in prison.
Oh no, not for the blind.
Yeah, I know. God damn. Why prisoners always be doing shit for us? We didn't ask for that.
Right.
The families of the victims opposed his release. 3 days later, the parole board refused his release. They instead decided to move him to an open prison. So he was moved to an unknown location to be in an open prison.
Okay.
On May 3rd, 2018, he was refused parole again. But in 2021, on June 7th, he was granted release on conditional license.
What does that mean?
He has to follow like certain. It would, I guess, like kind of like
Maybe it's like house arrest.
Yes, that's what I could compare it to. I would think is house arrest kind of.
So he's free.
We're getting there, but we're in 2021, June 7th. He's released. State Justice Robert Buckland put in a review to the parole board under a new board reconsideration mechanism. It's like a new law.
Okay.
They refused to review his case, which in this new law prompted him to be released on September, then officially released. So they decided that they're going to release him. They officially released him September 1st, 2021. Luckily, he was recalled to prison then on November 1st. So from September 1st to November 1st, because he couldn't stop talking to women and couldn't follow the rules.
Yeah, this is why we don't give these people chances.
He was literally out like September, October, 2 months.
2 months.
He couldn't control himself. But he's such a great, he was just, I don't understand. You know, he was just so perfect in prison. There was no women
Right, like,
to be talking to, and he just like.
And do they not understand there's a psychopathy behind crimes most of the time? Like, these people are putting on a show to get released.
Yes, of course. It's a game to them. That what do they have to fucking lose? They're in fucking prison.
Yeah. Yeah.
Yeah. A quote from Dawn's mother, quote, "He's been put away and women and girls are safe and protected from him now," end quote.
Good.
Secretary Dominic Raeb promised a parole board review, and Detective David Baker said he believes Pitchfork would pretend to be better to get out.
Yes.
Then it was in June 2023 now. He was released again on parole after a hearing.
When are we going to learn our lesson?
I don't know. So
women's lives don't matter.
No,
apparently.
they'd rather the man be comfortable. They don't care about us. So with public outrage, it forced Lord Chancellor to ask the board to reconsider, since Pitchfork couldn't follow the rules the last time he was out for only a few weeks.
Yes.
So after reconsidering in December of 2023, parole was denied, so he remained in prison.
Thank goodness.
The last update I have is from 2025, where Pitchfork requested another view to be released. He is 65 at this time, by the way. If the hearing was to happen, it was going to be live broadcasted because the case had gained so much attention. They had a preliminary hearing privately where they decided in October of 2025 to deny Pitchfork's release again. And that was the last.
If this man gets out again, we're all going to grab pitchforks. Yeah. And we're going to riot and chase him with our pitchforks.
Yes. Yes. Yeah, because fuck him.
That's what's going to happen.
Yeah. That was like, I know it's a little different over there, but damn.
What a wild ride.
Yeah. But hell yeah, DNA. I mean.
Yeah, hell yeah, DNA.
If our justice system could just, well, I mean, it's over there, but if the justice system could just fucking, when DNA does what the fuck it's supposed to do. Like,
But even here,
yeah,
our justice system needs a
it's
lot of work as
garbage.
well.
Yeah,
Yeah.
very, very true.
Another topic we talk a lot about in the true crime space, mainly in our true crime news episodes, is genetic genealogy. Genetic genealogy combines DNA testing with traditional record-based research to determine biological relationships, map ancestral origins, and build family trees. By analyzing shared DNA segments, like SNPs, it identifies matches in databases, often tracing ancestry back through many generations. Common applications include discovering birth parents, verifying research, or using investigative genetic genealogy, or IgG, to solve cold cases. There are 3 sources of DNA used by genealogists, which would be autosomal DNA, which identifies relatives from both parents within the last 5 to 7 generations. Y-DNA, like Amber talked about, traces direct paternal lines for males only, and mitochondrial DNA traces direct maternal lines. Autosomal DNA dilutes quickly and can typically only identify ancestors within the last 5 to 7 generations, like I said. Mitochondrial and Y-DNA tests are limited to relationships along a strict female or a strict male line. Mitochondrial and Y-DNA tests are used to identify archaeological cultures and migration paths of a person's ancestors along a strict mother's or strict father's line. A person's haplogroup can also be identified, which is DNA or chromosomal segments derived from a group of people who share a common ancestor. Our main focus obviously is going to be the IgG, investigative genetic genealogy, or also called forensic genetic genealogy, which merges forensic DNA analysis with traditional genealogical research to help identify suspects in violent crimes or unknown human remains by searching public genetic databases. By finding family matches to a crime scene profile, IgG builds family trees to narrow down candidates. As of December 2023, it has helped resolve over 650 cases. So
That's wow. Yeah, that's
a lot.
it is. That's awesome.
Hell yeah, DNA.
Hell yeah.
The process of IgG utilizes public direct-to-consumer databases and the same genetic genealogy techniques that have historically been used to help adoptees identify their families or parents. IgG involves determining relationships between individuals based on their shared DNA. When combined with traditional genealogy research, potential identifications of unknown individuals can be made to help in unidentified human remains cases or to develop leads in unsolved violent crimes. When DNA is collected from a crime scene, that DNA is uploaded into an FBI database called CODIS, the Combined DNA Index System. CODIS is populated with DNA profiles of both existing criminals whose crime meet the criteria to collect a DNA sample, and crime scene DNA profiles of unknown suspects. Once uploaded, the system will search the database to match either an existing criminal or additional crime scenes. If no match is found, the DNA remains stored in the CODIS database for future searches. It's at this point where investigators may decide to pursue IgG to identify a suspect. So what are the steps in this process? DNA extraction and profiling, like Amber talked about. Forensic scientists collect biological evidence, so semen, blood, saliva, skin cells, and use a high-density SNP single nucleotide polymorphism array to create a DNA profile suitable for genealogy databases. There's a database search. The DNA is uploaded to consumer-accessible databases, specifically GEDmatch, Pro, or Family Tree DNA, where it is compared against individuals who have opted into law enforcement searching. Then they identify matches. So genetic genealogists look for matches, aiming for close relatives, so like second to fourth cousins. The degree of relatedness is measured by centimorgans. Once they have a list of close matches, they see who in that list matches each other and who does not to form distinct clusters. And then they build a family tree. Genealogists construct family trees of the identified matches to find a common ancestor, often referred to as a union couple. Typically, a genealogist would start with the strongest match to get an idea of how many generations back they need to build a tree before moving on to the next cluster and finding a union. And then they do some descending research. So once the common ancestor is found, the genealogist works down the tree, researching all descendants of that couple to find candidates who fit the profile of the unknown subject or unsub. Target identification and confirmation. Potential candidates are narrowed down using additional evidence, like obituaries or records. Once a genealogist identifies a potential suspect, or a short list of possible suspects, law enforcement must lawfully obtain a DNA sample, which they compare to the crime scene DNA in CODIS. Only if there is an exact match can an arrest be made. Parabon Nanolabs is the most well-known company working in this field. By January of 2021, Parabon claimed to have used genetic genealogy to produce an investigative lead in over 200 cases.
Hell yeah.
So that's a lot in just 4 going on 5 years.
Yeah, and like we've said, it's a difficult process.
Yes. The DNA Doe Project, a nonprofit organization, have also been instrumental in resolving unidentified remains cases, many of whom are victims of violent crimes. Probably the most famous case solved using IgG that is considered the birth of forensic genetic genealogy is the capture of the Golden State Killer in April of 2018. And that was the former police officer, Joseph James D'Angelo.
Yeah, I never got to pay for his fine.
Yes. He was responsible for at least 13 murders and more than 50 rapes in the state of California between 1974 and 1986. That case went cold for over 40 years. Police were able to collect a DNA sample from him to confirm he was the right guy, and in 2020, D'Angelo was sentenced to 13 consecutive life sentences. Since 2018, when IgG really took off from this case, it has helped resolve over 1,300 cases, according to a study from December of 2025. So lastly, I'm going to share a case that was solved using investigative genetic genealogy on Barbara Blatnik. At the time of her death, Barbara was a 17-year-old high school senior from Garfield Heights, Ohio, a suburb of Cleveland, and went by Barbie. Barbie's sister Donna described her as a beautiful young woman and a bit of a free spirit. And her sister's quoted saying, "She loved music, AC/DC, Led Zeppelin. She once hitchhiked from Garfield Heights to Richfield to see Angus Young. She managed to work with her way up to the front row. She's one of those people who could walk into a room and connect with everyone," end quote. On December 19, 1987, Barbara Blatnik had plans with her friends. Around 6:30 p.m., she was picked up from her home by friends to go to a local bar that was popular in the area due to its lax policy on serving alcohol to underage customers.
Well,
Barbie was carefree and a bit of a rebel. She sometimes ditched school, smoked cigarettes, and enjoyed partying on the weekends. Barbie and her friends stopped at another friend's house around 8 p.m. to drink and hang out some more before eventually going to a small party at a local bicycle store within walking distance. She called her parents around 10 p.m. to tell them she would be coming home soon. Sadly, Barbie never made it home. Barbie's parents noticed she wasn't in her room the next morning, but this did not cause any immediate alarm because she would often sleep over at a friend's house on the weekends. Around 10 a.m. that morning on the 20th, the naked body of a young woman was found dumped by the side of O'Neill Road in Cuyahoga Falls. She had been beaten, raped, strangled, and dumped on the side of the road. Police noticed a class ring on the woman's finger with the name Barbara Blatnik engraved. Police showed up to the Blatnik family home where her father answered the door. He said his stomach dropped when he saw police standing there. He instantly knew something bad had happened because Barbie didn't come home the night before. The coroner speculated she was killed around 8 a.m., about 2 hours before her body was found. DNA was collected from under her fingernails, suggesting she likely fought off her attacker. The Generation Y podcast reported the coroner had severed Barbara's hand off and sent it to another lab. They noticed a phone number was written on her palm, possibly the phone number of a taxi, although it has never been confirmed, and apparently the hand has been missing since.
What the heck?
Yeah. Police questioned Barbara's friends, who reported they last saw her around midnight when they dropped her off at the corner of Warner Road and Grand Division in Garfield Heights, where her boyfriend Jerry Steed lived, but she apparently never made it to his house. According to the Porchlight Project, which is a nonprofit that provides free private investigative services, funding for DNA testing, and media support for cold cases who took on Barbara as their first case, they said it was actually easier to drop her off at the corner and let her walk over to Jerry's house as opposed to dropping her off right there because of the parking lot situation there. I don't really know what that means or what it looked like, but apparently it was easier to drop her off at the corner of the road.
Okay.
Police did question him, but he said he never saw Barbara that night. When if you think about it, like, that's crazy that someone saw her and took her in that short period of time where she would have walked from the corner to his apartment.
Yeah.
Like, what are the odds?
Scary.
Mm-hmm. At the time of her murder, her sister Donna was planning her wedding where Barbie was supposed to be the maid of honor.
Oh, that's sad.
That's really sad.
Yeah.
Like, it would be hard to even go through with the wedding after that.
Yeah.
Barbara's case went cold for many years until 2019 when the Porchlight Project reached out to the Cuyahoga Police Department. They were aware of how genetic genealogy was used to find the Golden State Killer the year before and suggested that police use that technique with the DNA found in Barbara's case, with Porchlight covering the cost. Because, one thing we didn't really discuss, it is very costly to do DNA testing.
Yeah.
And that's often why a lot of cases are still unsolved, because the funding isn't there to retest DNA.
Yeah.
So companies like this are very important in the true crime space. The Porchlight Project paid $6,000 to sequence the DNA found under her fingernails, which required a complex procedure to separate Blatnik's DNA from that of her attacker. Kind of what Amber talked about. A genetic genealogist matched the resulting DNA to a 67-year-old Cleveland man named James Zalstalnik. Police took a sample of his DNA, and in May of 2020, he was arrested and charged with Barbara's murder. Of course, when police first questioned him, he denied knowing her and boasted that he had many sexual encounters in his younger days and was quoted saying, "Too damn many of them. I used to be a whore," end quote.
Sorry. I laugh.
Police got wind of a jailhouse phone call he made to his sister where he says, quote, "I've been racking my mind over this crap, and I'm thinking maybe I hooked up with this girl way back when and we had sex from what I can remember. She was coming down Warner Road. I seen her walking and it was cold outside and I said, 'Hey, you want to ride?' She said, 'Do you live by here?' And I said, 'Yeah.' And she said, 'Can I use your phone?' So I took her over to the house. I put her on the phone and she got into a big argument with somebody and slammed the phone down and started crying. So one thing led to another and we wound up in bed. But the last I saw her, I dropped her at the corner of Grand Division and Warner Road. That's the last time I saw her. So that's why my DNA got all over her," end quote.
Oh, shut up.
Yeah, okay, sir.
Yeah, I'm sick of these people.
One thing led to another and you just wound up in bed? Yeah, okay.
Yeah.
This was not a consensual encounter, sir.
Yeah.
This marked the first time he acknowledged meeting Barbara. I'm assuming she must have been with him for a few hours before he killed her, if she was last seen at midnight, but the estimated time of death wasn't until 8 a.m.
I mean,
so it makes you wonder, did she willingly go with him at first and spend the night there?
That is possible.
And if so,
and then
what happened to make him kill her then?
Something just happened then.
What happened? Yeah.
I mean, he's obviously like a monster, but two people can have a consensual night together, and then what happened in the morning happened in the morning, but
Right, but I feel like she was going to see her boyfriend, though, so I doubt it was consensual. He probably was just holding her captive or something.
You're right. That's probably what is happening here.
And then he assaulted her, and then, clearly he kept her there for hours before he killed her. Maybe he just decided, I don't want her to tell the police it was me and killed her. I don't know.
Yeah. Maybe he just really didn't know what to do with her and just kept her for a couple hours till he decided. Yeah.
Yeah, I don't know. I can't imagine what that night was like for her.
No.
Unfortunately, or maybe fortunately, Zastolnik died of cancer before he could go to trial. Barbara's sister Donna was quoted saying, "I don't like the end of this story. Barbie didn't get her day in court. Our father didn't. I didn't. I wanted to look him in the eye and ask him why he did it. I wanted to call him a liar, and I wanted the whole world to know what a horrible man he is. He's a maniacal murderer and rapist, and he got away with it. He literally got away with it," end quote. And she's right.
Yeah. Yep, she's right.
So it's very sad. Like, I'm glad we know who he is and that his name is forever tarnished, but I mean, he didn't serve any time for what he did to Barbara.
No.
So I don't know. I mean, I guess we can accept him dying of cancer as a form of punishment, but it's not the same.
It's not the same. You're absolutely right. It's not the same. It's not justice. It's still not.
And then actually, just recently, I came across a very recent update in a cold case. The article said it is the oldest cold case solved with DNA, but when I fact checked that, there was actually one a little older solved recently as well in 2021. That's a little bit older than this case. I think that one was from like 1958, and the one I'm going to talk about is from 1964.
Okay.
So the second oldest case ever solved with DNA is that of Mary Theresa Simpson. Mary Theresa was 12 years old and was last seen walking home from visiting relatives around 6:30 p.m. on March 15, 1964 in Elmira, New York. Sadly, her body was found 4 days later in a wooded area about 7 miles from her home. Her body was partially hidden under 4 heavy rocks. Her mouth was stuffed with dirt and twigs, and she was determined to have died from asphyxiation and was sexually assaulted.
Ugh, God.
In 2003, 39 years after her murder, DNA collected from semen on her skirt was entered into a national database, but there were no hits at the time. Investigators tried again in 2014, but still nothing. In 2022, a grant was awarded for DNA testing in the case. A 0.4 nanogram DNA sample, an amount too small to see, was sent to Othram Technologies for analysis. So Othram is a leading Texas-based forensic DNA lab specializing in forensic genetic genealogy to solve cold case murders, disappearances, and unidentified decedents. They have helped in numerous cases. Their website says, quote, "Othram Technology enables the National Missing and Unidentified Persons System, NamUs, and the Australian Federal Police's National DNA Program to identify human remains and resolve missing person cases. Local, state, and federal law enforcement agencies across the United States and internationally have partnered with Othram to break through previously impenetrable forensic DNA barriers and close previously unsolvable cases," end quote. So Parabon and Othram, there are two big ones that we hear a lot in cases being solved by DNA.
Yeah.
They also showcase cases they have solved on their website, so I highly recommend anyone listening to this to check out their website. Othram was able to upload a DNA profile to 2 DNA databases, and the FBI used it to build a family tree and identify a son who had submitted a DNA sample. His father was a truck driver named Raymond Murray, who had already died in 2004 at the age of 72 and was buried in Elmira. The National Center for Missing and Exploited Children, NCMEC, covered the cost and provided a forensic anthropologist to exhume Murray's remains so they could get a DNA sample.
Wow.
Authorities announced on February 10th, 2026, that the DNA matched.
Hell yeah. Hell yeah.
Mary's killer was 33 years old when he committed the crime, and he was also found to have a criminal history involving children, but he was never a suspect at the time. So fuck that guy.
Yeah. Do we know if he spent some time?
I don't know if he was ever arrested
or anything.
Time for his crimes, but yeah, he was just a truck driver, and genetic genealogy called him.
Wow, that's crazy.
Yeah.
Sadly, I feel like there's probably more cases than that than you would think with the truck driver.
Right, yeah.
Especially back in the day.
And it's a lot easier for them to get away with crimes, too. They're just sleeping here for a night, and then off they go.
Constantly. They can literally just be sleeping in their truck, not even spending money on the road.
Mm-hmm. Yeah. Mm-hmm. Yeah, I never trust truck drivers. No offense.
No, this.
But
I'm not
I
getting
don't
any
like to
trouble.
soften at rest stops for this reason either.
Yeah, yes, I hate rest stops.
Mm-hmm. Yeah, and if it's just me and Brandon, I'm not using that bathroom.
I'll
If
do what
I'm
I
with
have
a
to do.
female or something, then we'll go together. But
Oh yeah,
otherwise, I'm not using the truck stop bathroom.
yes.
So that was the 2 cases I have for genetic genealogy.
Hell
Solving
yeah,
them. Yeah.
that's good.
So we hope you enjoyed this episode. Like,
Yeah, I hope. I know this is probably an episode you put on, especially when you're just like mindlessly doing something. I get it, but
mm-hmm.
I hope
Yeah,
today you dove in head first to be a little bit more knowledgeable about DNA with us. You know?
yeah. I hope you learned something.
Yeah, and help us get
And we
t-shirts.
threw true crime within it, so you heard a few cases in this episode as well.
Yeah, and help us with t-shirts ideas. Hell yeah, DNA. We did.
Yeah, yeah. I'm telling you, once we get merch, we're making a Hell Yeah DNA T-shirt
Yes, we are. And I want like a cool
and
little
a sweater
DNA.
because I always wear sweaters.
Yeah, you do. She's always cold. But with a cute little DNA guy or something on it, like some kind
Yeah,
of
yeah.
animation. Hell yeah.
Yeah, I always think of from Jurassic Park,
I know.
that little DNA guy.
Yeah. Try to give you educate you.
Yeah.
Yeah. Thank you for watching. Please give us a thumbs up if you like this video and check us out on all of our other social media platforms.
Yes, and please subscribe and turn on the bell to be notified of future uploads. And keep looking around for our next video. Bye.